So that concludes our understanding of different types of fermentation process. Okay, batch, fed batch and continuous. What is the similarity, what is the difference between batch, fed batch and between batch and continuous. Now we are going to talk about the third topic of unit 12, and that is plant tissue culture. So when you talk about the plant tissue culture, we want to understand first of all what is tissue culture. The plant tissue are totipotent in nature. The meristematic tissues of the plant, which are the growing part of the plant cell, the apical part of the plant cell, they continue to grow, they can grow. So if we take those cells, if we extract cells from there, and if we place them in a medium to supply them with proper nutrients and in an optimum environmental parameters that includes pH, temperature, oxygen availability, light source and everything, then those plant cells can grow a healthy adult plant. That's a possibility, and that happens. So here you can see a small plantlets is plated even in a culture tube, and after a few days you will see the plantlets start coming out. Their roots and shoot structures are visible. So simple process says the tissue sample can be taken from the plant source. That can be any place. That can be root tip, shoot tip, leaves, okay, and also parts of the flower as well. So different parts of the body. If you take it and tissue samples placed in an agar growth medium containing nutrients and also containing growth hormone, that is auxin. Okay. Primarily we need to provide them with auxin as a growth hormone, as a growth regulator, because obviously they need nutrients, and we can make the medium liquid or we can add agar to make it solid, but we need to provide the nutrients. The nutrient means what? Carbon source. We need to provide carbon source, nitrogen source. Apart from that, there is a growth regulator, that is a hormone auxin needed. After we provide them with everything, then the samples develop into tiny plantlets. Small plantlets are developed, and from these plantlets bigger plants can be produced, and we can then shift it to the soil and they can grow. So without soil, without even sunlight, with artificial light, without soil, if they receive proper nutrients, provided all the environmental factors and optimum value, plant tissue can give rise to a healthy plant. This process is known as micropropagation. Okay, how exactly? If we take a single part of the cell from plants and we can grow into different plant cells, different adult plants from that one tissue. Now the culture process, this is called the tissue culture, plant tissue culture or anything. So plant tissue culture process requires first of all we need an explant. What is an explant? The tissue we want to culture, and this explant need to sterile this, right? And we also need to sterile the medium, and then you put the plant or explant into the medium. Now when we say medium, what do we have in the medium, right? What do we have in the medium? Nutrients, right? Nutrient means carbon source, and apart from the nutrients or carbon source, we also need temperature, pH, light, salinity, and we need growth regulators, right? Growth regulator is auxin, and the nutrients, among nutrients we have macronutrients and we have micronutrients, right? We'll see different type of nutrients that are used. One is the carbon energy source, that is the carbon source, sugar. But apart from sugar, there are also other nutrients necessary for a plant's growth. We'll see that in a moment. So many plant cell cultures, as they are not photosynthetic, also require the addition of fixed carbon source or sugar. Apart from sugar, sometimes you also need to add organic supplements such as amino acids and vitamins where they have rich nitrogen in it. So these are nitrogen sources, amino acids, vitamins are sometimes needed for the enzymes to work properly, and sugar is as a carbon source, and then other physical factor, temperature, pH, gases in the environment, light and osmotic pressure. Now here we are going to talk about the different type of uh, nutrients that we are going to use, okay, in a medium of plant tissue culture. And when we say that, it's not only about plant tissue culture where the plants need these nutrients for the growth. In real life, plants that we are seeing growing in the soil also requires these nutrients for the growth, right? So this table is very important where we discuss about the importance of different nutrients for the plant's growth. You need to understand and remember this table. Also there is another table, deficiency of these nutrients and what are the diseases that form in the plants. These both tables are equally important. So the first thing that I'll say is the name of the different types. So primary, macro, remember I told you there are macronutrients which are primary nutrients, predominant ones. There are micronutrients. Micronutrients are also necessary, but without micronutrients the plant will not die. But there are some physiological problems visible from outside, and also you can see that in the impact in the plant cell. But without macronutrients a plant will die, will not survive. So the primary macronutrients are three different types: N, P and K. Nitrogen, phosphorus, potassium. N, P, K. And generally this nowadays is produced as a mixture of NPK. We call it different names, but the NPK is present in a particular ratio. In this case the percentage in the plant nitrogen is known as 100% plant nitrogen, comes from phosphorus 6%, potassium 25%. What is the function? You know, nitrogen is needed for protein and amino acid synthesis, phosphorus is needed for nucleic acid ATP synthesis, and potassium for catalyst and ion transport. Secondary macronutrients. So these are primary macronutrients, these are secondary macronutrients. Still it's needed, but secondary macronutrients. Calciums, cell wall component. Magnesium, part of chlorophyll structure. Sulfur, producing amino acids. Iron, part of chlorophyll structure, okay, and required in the chlorophyll synthesis. So magnesium is a part of chlorophyll structure and iron is needed for the synthesis of chlorophyll. And what is the total relative percentage is plant to nitrogen? Nitrogen is 100%, but relative to nitrogen how much percentage the rest of them are present? Means the percentile values with nitrogen that we are checking here is that potassium is near to the 25% of the total nitrogen, 12.5 is calcium, okay, then 8 is magnesium, 6 is potassium, phosphorus, 3 is sulfurs, and iron is very low, 0.2. Now we'll have last thing, that are micronutrients, which are needed in very tiny amounts for the proper functioning. Most of these micronutrients are components of enzyme activity, like the cofactors that we know. There are different micronutrients acting and helping in the enzymes to perform their own function. Copper, manganese, zinc, boron, molybdenum, chlorine, these are all part of micronutrients and they are part of the enzymes. See, copper part of enzyme, manganese activate enzymes, zinc activate enzymes, boron cell wall component, molybdenum involved in nitrogen fixation, and chlorine helps in photosynthetic reactions. Okay, so these are the list of different, and you see the micronutrients, why we call them micronutrients? Because they are present in very low amount, their activity is there in very low concentrations, very low concentration compared to the nitrogen. Okay, now we are going to see the second important table, and that table is regarding the diseases. Okay, if any of these macronutrients are missing, then what problems the plant will face? Because when you are culturing the plant in a plant tissue culture lab, you need to provide them with proper nutrients, and if any of these properties are visible from outside, you can promptly understand the problem with it and you can fix it, right? So what we have here, we have these issues. For a new growth, there are two situations. One is the new growth, other one is the old growth. There are difference between the two. New growth means where the growth of the plant tissue, new plant tissue coming out, that is apical bud of the shoot, root ends and all. These are new growth. And old growth is when there is, let's say, a stem is somehow broken, a new stem is coming out from the old region, that is old growth. So in the new growth, we have two different deficiencies. One is the iron deficiency, other one is the calcium deficiency. The iron deficiency cause young leaves yellow and white with green veins. Okay, mature leaves are normal. So if iron deficiency is present, the very young leaves will be yellow in color, but once they mature, they will be normal color. That's the deficiency of iron. If there is a deficiency of calcium, the new leaves mishappen or stunned. Existing leaves remain green. So they will be like new leaves will not even form, or even though they start forming, they will be stunned. They will not be a proper shape or size. For the old growth, rest of the other deficiencies are visible. So nitrogen deficiency, what we see, the upper leaves are light green, where the lower leaves are yellow, bottom all leaves are yellow and shriveled. This is what the nitrogen deficiencies look like. Potassium deficiency, potassium deficiency. Yellow at the tips and edges, usually in younger leaves, dead or yellow patches developed on the leaves. Yellow patch developed on the leaves. Phosphate, leaves are darker than normal and loss of leaves. Okay. Carbon dioxide, white deposits on the leaves, stunned growth of the leaves. Manganese, yellow spots or elongated holes between veins. Magnesium, lower leaves turn yellow from outside going in, veins remain green, but the leaf turns yellow, mainly the lower leaves. So these are all the different symptoms of major and macro and micro elements for the growth, or macro and micronutrients for the growth of the plant. Now what are the different types of cell culture or tissue culture that we do? There are different types of tissue culture. As I told you earlier, that the plant tissue, if you take it from apical bud, generally we take it from apical bud, from the growing tissue, for the new growth. If you look at the plant growth, any part of the plant cell which is under the new growth, we take the tissues from there. It's better to take tissues from there because those tissues are actively dividing tissues, actively dividing cells, and they can grow faster, right? And one more advantage to take cells from the apical tissue is that their growth is so much fast that the viruses or any pathogen cannot cope up with the growth of the tissue. That's why the apical tissues, if you take them, they are generally sterile in nature. You don't need to think of extra sterilization of the tissue because they are generally sterile. So this is apical, you can see this is lateral bud, this is apical bud, but both are meristem tissues, and you can take the tissue from there. So this is wild plant, okay, and then what we can do is simply we can transfer it to the tissue, the culture media, allow it to grow. Or what we can simply do, we can call it normal growth, or sometimes we targetedly insert a gene of interest, make a recombined cell and then allow the recombined cell to grow. Generally we can do that with agrobacterium, right? Agrobacterium is a natural gene delivery system, natural gene delivery vehicle to donate or transfer target DNA, target gene inside a plant cell. And there are two different species, Agrobacterium tumefaciens and Agrobacterium rhizogens. Agrobacterium tumefaciens infection cause shooty teratoma lines, and the hairy root lines means basically the Agrobacterium tumefaciens cause root nodule formation like this, tumor formation in the root nodule known as crown gall. So in this case we are focusing on the medium free of hormones and it continues to grow. Similarly in Agrobacterium rhizogens it forms hairy root syndrome. Again the root will be much hairy. So hairy roots lines, and again we can use it for the screening and for the production and growth. But this is one side thing, like after extracting the wild type plant we can infect it with Agrobacterium tumefaciens or rhizogens. But that's an optional idea. That's even complicated form where we extract the cell, we transform, then we allow it to grow and culture. But in normal mode we simply take out the plant cells and place it to the media with proper nutrient, and you place it to the proper environmental parameters, it will grow. On the other hand, the wild type plant can produce what is known as callus in the process known as callogenesis. Callogenesis is the process of callus formation from wild type plant. So from wild type plant callus is formed, okay. Direct or in vitro organogenesis is done, and in this direct vitro means what? We have medium composition and hormonal balance need to be provided, and then untransformed shoots or roots, whatever things are present which are not transformed, okay. So in this case we are simply seeing whether the transformation is done or not, but without the transformation also we can simply take the cells, put it in the medium, and once we put the cell in the medium, cell can form callus, it may not form callus. So callogenesis can be achieved, may not be achieved. We'll see in a moment that in some cases we can achieve callus formation. Remember, in case of plant hormones, we have seen the role of auxin and cytokinin. The ratio of auxin and cytokinin plays a vital role to dictate whether the callus will be formed or not. If the ratio is proper, the callus is formed, then from the callus the plant can grow, and sometimes without forming the callus also the plants can grow, okay. So in both this case one thing is common, that is we take the explant, we take the tissue, we put it into the culture media, allow it to grow in the environment, it will grow. Now based on that we have shoot tip culture, root tip culture, callus culture, anther culture. There are different modes of culture. But what we need to understand for CSIR NET, where the CSIR asks many questions every single year, and that is regarding somatic embryogenesis.